Good morning, good afternoon, good evening to all of you connected with us from China, North America, South America, Australia, and of course Europe. Many different universities, research centers, and single persons interested in neuroscience are connected with us now. It's really my pleasure. I am Giancarlo Comi, Honorary Professor of the University Vita Salute San Raffaele here in Milan. It's really my great honor to introduce you the Human Brains Project. So we know that the human brain is the most complex biological entity in the known universe. I think that everybody will agree with this point. How thoughts, emotions, perception, individual and social behaviors can be generated by this relatively small organ is still largely unknown and subject of very important studies. It is also important to notice that the brain function analysis is fundamental also in the context of neurological diseases, so relevant, and particularly here I want to mention neurodegenerative diseases. And this is really what matters in order to try to develop new treatments for still not treatable conditions. Human Brain is a global project of Fondazione Prada. The project was born in the mind of Miuccia Prada. I want to mention here what she said to me when we met the first time. This specific project dealing with science is a true challenge for me. Perhaps the most important for Foundation Prada so far. Our approach will be multifaceted, multidimensional, multi-disciplines will be involved, and it's fundamental to combine together these different aspects in order to face the complexity of the brain and the brain function. So, professors of psychology, philosophers, bioengineers, neurophysiologists, geneticists, and many others will be in some way involved in this section. Human brain's activities will consist of different types of activities, exhibitions, scientific meetings, workshops, public debates, and editorial activities. So educational aspect will be really fundamental for us. A team of people you see here presented have been fundamental for the production of this project. It is about a couple of years that we are working to this project. I want to start with Udo Kittenman, the former director of the National Gallery in Berlin and many other directions of exhibition in his history, and curator of many different exhibitions, for example, the Venice Biennale. Massimo Cacciari, a philosopher, well known everywhere. Daniela Perani, a neuroscientist, neurologist, and a great expert in the area of neuroimaging. Andrea Moro, deeply interested in the area of language, as it is Jubin Abutalevi, particularly Jubin is interested in multilingualism. And last but not least, Viviana Kazam, a journalist for neuroscience, and also much more than that because she founded very important initiatives in neuroscience such as Brain Circles. All these people have worked together having in mind some very important goals. The experiment with new waves of dialogue between scientists with different qualifications and with those who are not really professionally involved, but they are curious to know about brain and mind. It was also the way and the appropriate approach in order to test innovative communication formats towards an heterogeneous and international audience. It is a challenge, but it's something fundamental. We want to create a convergence of distant scientific and humanistic perspective and approaches to come up with a general synthesis that would be of great value for everybody. Integrate the rigor of scientific knowledge with the cultural work and communication potential of Fondazione Prada. Decline the brain to the plural. You see, we speak about human brains. And human brains is fundamental because no one brain is similar to another brain. We have much more variability than our fingerprints. So it's something unique in the history, unique in the past, present, and future. And this singularity is really one of the key aspects. We have three major organizations and events that we want to have in these 2-3 years of activities. First of all, the Culture and Consciousness online conference, the one that is just starting now. Then next year, we will organize a very important meeting on neurodegenerative diseases. This is an event co-organized by some of the major research centers in the area of neurodegeneration. We want to bring together not only scientists, but also those who are at different levels involved, including those who have responsibility of political decisions, and single persons, ethicists. And finally, we have the plan in 2022 to participate at the Biennale of Venice with an exhibition on brain. Again, a quite strong challenge for all of us. Well, we are here now because we start with the first discussion. It will be the first of 5 daily live stream discussions. We use this definition of discussion because we want to give the opportunity to listen to some of the more relevant, important, and famous researchers in the area of neuroscience, and particularly in the area of consciousness. The title, Consciousness and Culture, is telling us, and remind everybody, that is the consciousness, the basis of the culture. Without consciousness, there's no culture. And please look here, you see that all the activities will take place every day at 7 p.m., like this one. And here you see the connection you might have in order to participate to the event. I want just very shortly review with you the other type of discussion. There will be a second discussion with Yves Marder and Antonio Damasio, moderated by Daniela Perrani. I will give you more details about this one later. Then we have a third discussion that will be in the area of language, something exquisite, human characteristic. And the fourth discussion will be based on the evolution of culture, how far we will go. This is one of the key questions. Is there the time now to have machines able to have consciousness? And finally, the 5 days will be closed by the discussion 5, where a philosopher and a very famous neuroscientist, philosopher is Michele Di Francesco, and Giulio Tononi is the neuroscientist, will really discuss from different point of views the topic of consciousness. And Massimo Cacciari and myself will at the end take some concluding remarks. Before that, I give the role to our magic speakers, I want just to concentrate on some general concept that you see listed here. Consciousness is really a subjective experience. Cogito ergo sum was the famous sentence of Descartes. So, what is it like to be able to perceive a scene, to see a color, to recognize a face, to renting a thought, or to reflect on the experience itself? These are all magic aspects of the consciousness. And here what we will try to do is to go behind the scene, to try to understand which are the mechanisms that are really able to produce consciousness. Still quite far to be fully understood, but as you will see, there is a lot of advancement, particularly in the last 15, 10, 20 years, due to some incredible developments in the area of neuroimaging, of genetics, and of biology. So, it's now the time to go directly and to introduce our two first speakers of today. We have with us Mavi Sanchez-Vives from Barcelona, Spain. She will talk about the research for consciousness, brain processing of real and virtual worlds, and Jean-Pierre Changeux, let me say a giant of neurosciences. He will talk about the connectomics of conscious processing from the molecular to the cultural level. So, maybe really welcome here. A great pleasure to have you with us for the just onset of our activities. It's really a privilege to have you with us. I think that your contribution along these years in the area of research has been really fundamental. Mavi Sanchez-Vives is a neuroscientist at the ICREA Research Professor and at the Institute of Biomedical Research, Auguste P. Einsteiner in Barcelona, where she is also the head of the System Neuroscience Group and co-director of the Event Lab at the University of Barcelona, where also she is adjunct professor of the Department of Basic Psychology. So many different activities, but they really integrate in the same person different approaches, and this is really one of the characteristics of Mavi. Mavi is also deeply involved in the Human Brains Initiative in Europe, where he has the responsibility of one of the work packages. Mavi, it's really again a great pleasure to have you with us. It's now up to you. Please.
Okay, thank you very much for the introduction. It's a great pleasure to open this series about human brains, and I want to thank the Fondazione Prada for the initiative and for the invitation. And being a neuroscientist, I have to say I think this initiative is quite timely. So if the coronavirus would have not altered and interrupted our lives a few months ago, I would have said that we are starting what we could call the age of the brain. So why would I say that? Well, I think there is a renewed interest in the brain coming from many different fronts. So I will mention 4 of these fronts. On the one hand, from the governments, we can see that there are initiatives going on, very largely collaborative projects, not only in Europe with the Human Brain Project, of which I'm part, and in which there are almost 100 institutions in Europe collaborating, but also in other countries. There is in the United States, there is the Brain Initiative, there is the China Brain Project in China, in Japan there is MINDS, in Korea there is Brain Canada, Australia, Israel. So there is a moment in which there is an interest for the governments to advance in the study of the brain. On the other hand, I can see that there is a very large interest from the side of the general public. So people are genuinely interested in understanding about their brain, with the idea of each one has different motivations. People want to improve their brain function. They want to prevent neurodegenerative diseases. They want to understand the science of the brain, what the brain has to tell you about different aspects. And this is why I have selected in this slide different books that appear. Some are more scientific than others, but they respond to this demand, to this interest. And there are books on neuroeconomics, neuroscience, neuromusic, neuromarketing, neurogastronomy. So it reflects this interest from the general public to try to understand as much as possible about their brains. On the other hand, we see a new interest from corporations. So there was recently a very announced intervention of Elon Musk presenting Neuralink, his brain lab. But there is also similar initiatives from Apple, from Google, from Facebook. They have now neuroscience labs. They have labs to study the brain. And finally, if we look in the entertainment world, there is, here I included some images, but there is a very large interest also, and this awakes the interest in the people. So there is chapters about brain interfacing, how to tap into the brain, about dreaming, consciousness, and so on. So why is this happening now? So from my point of view of scientist, I think there is two reasons why we should be interested now. And these reasons are that we can study the brain like never before, and that we need to study the brain. There is this need as well. So we can study the brain like never before because there has been many advances. The study of the brain requires from advances in many different disciplines. So we have seen in the last 20 years, 30 years, advances in brain imaging, in optics, in micro and nano electronics, in new materials, in light control drugs, in optogenetics. Something very important, which is the computational capabilities. We can nowadays acquire huge amounts of information and analyze these very large amounts of information. There are also developments in robotics, in virtual reality, in genomics, gene therapy. So we have all this array of tools that were not available before, and they allow us to dig in how the brain works. But on the other side, we really need to study the brain. And we need to study the brain because there is a very large prevalence of neuropsychiatric diseases. In Europe, there is a rough estimation of 180 million people per year. This prevalence increases with the aging of the population. It affects the life loss due to early disease or disability, and it has a huge economic cost of roughly 800,000 million euros per year in Europe. So there is obviously a very important need. And of course, there is the need of us as humans to try to understand ourselves. So when did this started? Well, for the last 2,000 years or more, humans have been trying to understand the solar system, the universe. However, the in-depth study of the brain has a much shorter history that we could say that with some detail started around 150 years ago. And there was these studies carried out by two men that we can see here, Camilo Golgi and Santiago Ramón y Cajal. They both used this method developed by Camilo Golgi of silver staining. And we can see here some of these reconstructions. And they both share a Nobel Prize in 1906, even when they have theories that were to some extent contradictory, the neuronal theory and the reticular theory. However, even when neurons happen to be independent elements, as I will mention today, we will see that really the brain functions as a very large network. So they were some of the first people that were able to look into this structure that contains what we are, what we plan, what we think, and to look into it and to be amazed by these incredible structures by these neurons. These are some, in this slide, you can see some of these drawings made by Cajal of different structures. Here, the cerebral cortex, or the retina, the spinal cord, Purkinje neuron in the cerebellum. And if we look in a bit more detail in the cerebral cortex, we can see these dendritic spines carefully drawn where the inputs, the synaptic inputs arrive. And we can see also these very interesting arrows that Cajal drew where he suggested, without any real measures, how the information would flow in these branches. And I have this here, and I'm going to show to you some animation shared with me by my colleague, Javier de Felipe, where you can see this kind of neuron and reconstruction as it can be seen today. Here is some spiny neuron that he has filled, and you can see here on the right these beautiful dendritic spines that have around 4 micrometers, and we can see very well, and it's going to receive these synaptic inputs arriving into the cells. These circuits in the cerebral cortex are formed by neurons. Here you can see a pyramidal neuron, and they receive inputs, afferences, information also from inhibitory cells. Here there are 3 of them represented. So so this forms a universe. So often the brain is compared with the universe because it's so large. There is 86 billions of neurons. There are 10,000 in humans, even 30,000 inputs per neuron. This means every neuron integrates information from even 20 or 30,000 other neurons and 2 or 3 million kilometers of connections. So it's really like a universe that contains ourselves. And as it has been said, if our brain would be simple, we would be too simple to understand it. So we are complex, and our brain is also very complex, and that's why, right? So we are now going to have a look inside this cerebral cortex. This is a reconstruction made by my colleagues in the Blue Brain project in Madrid, shared also with by Javier de Felipe, and I think you should be seeing it by now, digging into this network. And I think the video is being run, so I hope you are seeing it. And I would like to make 2 points here. One is that you can see this incredible complexity of the cerebral cortex. So as I said, each one of these neurons receives an incredible amount of information that is being processed. But also what is fascinating, if you look into this structure, is that this is the basis of what we are. This is going to generate a sentient being that is conscious. So what is a consciousness? This is what this session is about. And I'm going to speak about 2 aspects of consciousness. One is about consciousness levels. So what is consciousness when we talk about consciousness levels? So what it is what we lose when we fall asleep. So every night we close our eyes, we fall asleep, and then we disappear, we vanish. And the same happens, for example, if we get a few drops of an anesthetic in our blood. We just, what we are, it disappears, and the next morning, or when the anesthetic washes out, we come back to the world. So the brain can have, and this is what you can see here in this scheme made by Stephen Loris. So what you can see here are different brain states that can be sustained by the brain. So what it represents is wakefulness and awareness, and you can see that we can be in deep sleep, as I mentioned, or under general anesthesia, or we can be awake, or we can be dreaming. But there are also some pathological states, which are disorders of consciousness, and there can be a minimally conscious state or an unresponsive state where the person is awake but is unresponsive, which used to be called vegetative state. And there can be also a coma. So the brain network sustains all these different possible states that have different patterns of activity, different biomarkers that we are going to be interested to understand, to be able to identify, for example, whether a person that is unresponsive is conscious or not. So in a way, we can think that there are many brains in one brain, because as we will see, the brain is going to express different activity in these different states. So this is very relevant from a clinical perspective, because there are disorders of consciousness, as I mentioned, and there are many patients in this state, and these are numbers from the United States, and there are 50,000 unresponsive patients and 300,000 patients minimally conscious currently in the United States. These numbers can actually increase because the care in intensive care is better, and therefore more people survive. So it is very important to develop measures to quantify whether a person that is unresponsive is conscious or not, to what extent is conscious. If this person is conscious, provide them with means for communication, and also eventually design methods in order to bring their consciousness back. So for all this, we need to understand the basis. We need to understand the underlying mechanisms. So let me start by unresponsiveness. And this is here in this area in red. And unresponsiveness is associated to a very synchronized patterns of activity. And you can see here this activity, very slow oscillations where the whole brain discharges or has these synchronized patterns of activity. This was, in fact, some of the first activities seen in the brain. In the first recordings that took place were done by Catton in 1875, and this was the first activity that called his attention. And also in 1929, in the first electroencephalographic recordings in humans by Professor Hans Berger, was also some of the first activity, because it has very large amplitude. And it has very large amplitude, this activity that is associated to a slow wave sleep or to deep anesthesia. It has large amplitude because it reflects that there are many neurons active at the same time. And the cellular bases were not described in some detail until 1993 by Mircea Steriad and his collaborators that describe in more detail this pattern, but still the final mechanisms of how the red generates these emergent activities is still under discussion. So when does this activity appear? Well, as I mentioned, it appears in deep sleep, and I showed you it's here. And also we can observe it not only in the spontaneous activity, but also if we stimulate the brain, one can see that there is this type of response, this kind of biphasic response, which is, it doesn't propagate a lot and is very similar, we can say, in deep anesthesia. Interestingly, this activity also appears when there is isolated areas of the cerebral cortex. So if there is an experimental cut of a piece of the cerebral cortex, there is still the generation of this slow pattern, a slow rhythm. And this is the case also when there is a lesion. Here you can see when there is a lesion that disconnects some area of the cerebral cortex, one can observe this slow or delta rhythm over this disconnected area. In fact, quite a while ago, 20 years ago, we found that actually, even if we take a small piece of the cerebral cortex and we put it in vitro in a dish, it contains enough circuitry in order to generate this activity that you can see at the bottom of the slide, which is also a slow oscillation and is very similar to the one that one observes when there is the full brain under a slow wave sleep. So what this is telling us is that a small piece isolated of the cortex contains enough circuitry in order to generate this pattern of activity. And this is the case, as you can see here in this drawing by Lorente Denot, because there is recurrency in the circuits, and you will hear more about this during this talk. There is recurrency, some neurons, they connect the others and back, and there is a recurrency, and therefore there is in a small piece, there is enough circuitry to generate this activity pattern. And we suggested that actually this slow rhythmicity associated to unconsciousness is actually like a default activity pattern of the cortical circuit. And we have suggested this for all the reasons that I show, because in any circumstance, you isolate a piece of cortex and this activity is generated. And indeed, the same is the case in computational models. It can be very complex computational models, where many neurons are reconstructed, as we can see here in this work by Markram et al., that these neurons, they reproduce a piece of the cortical network, and they generate these slow oscillations. And the same is the case, as you can see here on the right, in this mean field model, basically a much simpler model that contains basically two elements, which is recurrency of the connections and adaptation. So in a way, we can say that the default state of the cortex is to be in a sleep, to be unconscious. However, obviously our experience is that of being conscious. So how is this transition from unconsciousness to consciousness? And it's very important to understand unconsciousness to see how it can be transformed into consciousness. So how is this transition? Well, if we look at an individual neuron, as this one recorded here by Steriad et al., what we can see when there is this transition from a slow wave sleep to awake, we can see how this rhythmic pattern stops and it gets transformed into a more or less continuous pattern of firing that is not rhythmic anymore, and it's to some extent asynchronous. So if we look at this activity, not spontaneously, but following an stimulation, and this is work by Marcello Massimini and collaborators, if one stimulates the cerebral cortex, there is, in these situations, in which there is unconsciousness, like sleeping, anesthetized, or unresponsive wakefulness syndrome, there is, in all the different locations, there is this very similar synchronized response, relatively synchronized response, this sort of biphasic response, which is associated to unconsciousness. While when there is a conscious state, like awake or even dreaming and so on, then there is this response that, as you can see, is not the same anymore in every point, but is different in the different locations. So this is the spatial aspect, the spatiotemporal aspect. And to which kind of networks does it respond? Well, in order to have this kind of more simple synchronized responses, we could imagine a network which is very local, is very segregated, or it can be all very highly integrated. And in these cases, we could obtain this kind of responses. However, the situation is different in the awake state, and there is this balance between these two, between these very segregated and very integrated. So this generates a high complexity in this network, which is associated to consciousness. So this is based on ideas originally proposed by Tononi and Edelman in 1998 in a paper in Science, and also the integrated information theory by Tononi is very connected to these ideas. So consciousness is therefore associated to these interactions and between different areas, causal interactions that have given rise in a practical aspect to one measure of consciousness, a measure that has a clinical value, and you can see it here by Casali et al., and it has clinical value because it can be done in patients, like I mentioned to you before, patients that are unresponsive and that one needs to know whether they are conscious or not. And this measure consists in providing a stimulation to the brain, to the cortex with TMS, with transcranial magnetic stimulation, and it evokes responses that are recorded in different locations. And I won't go into the details of how this is calculated, but here you can see in the space and the time of this response, so how the response is on time over many different locations in the cortex. And this is compressed with a Lempel-Sieve algorithm, this response, into one number, which is an index, which is referred to as the Perturbational Complexity Index. And this index has a relation with the consciousness level. So you can see here how in situations where there is an unresponsive wakefulness syndrome or a minimally conscious state is lower, and in an awake person is higher. So you can see on the right a segregation between these indexes. So therefore, this is a measure of how the response is in the many different areas of the cortex to a stimulus, and is giving information about the functional state. So therefore, from the point of view of the levels of consciousness, just to recap what I told you, in unconsciousness, there is, on the left, there is a high synchronization. There is low spatiotemporal complexity. there is a dominance to some extent of local interactions, and there is low interareal causal interactions. However, on the right, in a conscious state, the temporal pattern is asynchronous. There is a high spatiotemporal complexity, is more dominated by long-range interactions, and there is a high interareal causal interactions. So how about the neural correlates of consciousness? This was the name given originally by Crick and Koch in the search for what is the basis for consciousness. And this could be patterns or it could be neurons. So are there any specific neurons causing or controlling consciousness? Well, not that we know of, but we have seen that actually consciousness is about interaction between areas, and there are many neurons that are critical in connecting different areas in the brain. And I have here some examples to show to you. These are very long axons of pyramidal neurons that you can see how they have a very large distribution connecting very distant parts of the brain. We also have, and very importantly for consciousness, thalamocortical neurons. Some of them have also axons, as you can see here, that connect different areas, in this case motor areas, somatosensory areas. They are the axons of the same neurons in the thalamus that innervate separate cortical areas. This is an example of a claustrum neurons that were described by the Allen Institute, where these neurons, they have also connections around very distant part of the brain. And very important also, of course, in order to go from sleep to awake, is the reticular activating system, that all the neurotransmitters that are going to mediate in physiological conditions this transition, and they have also a very diffuse innervation of the brain. So with this, I'm going to move on to another aspect I want to talk to you about of contents of consciousness, which refers to a different aspect, which is contents of consciousness. So we have seen we need this interaction across areas to be conscious, but what are we conscious of? So when we think about the consciousness of the self, usually one considers that what are we conscious of? We are conscious of the external world, we are conscious of our own body, and we are conscious in our thoughts of ourselves, our history, autobiography, past, and also future plans, and so on. So let's talk about the sensory representation of the world. So, okay, so if we see this, okay, so the information, the visual information arrived to the eyes, is transduced into spikes, and these spikes travel to the thalamus and they arrive to the cerebral cortex. And this video that we just saw is how you can hear the spikes of a neuron in the primary visual cortex discharging. These are old videos, as you can see, by Hubel and Wiesel, that were the first ones that characterize this type of responses. These are single unit recordings. I'm going to play one. Okay, so this is what happens in the primary visual cortex. The information arrives and is encoded into spikes. Okay, and here I used to work in sensory processing, either in in primary visual cortex and also in auditory cortex. Here you can see these spikes, but now intracellularly recording from inside, with an electrode inside one of these neurons, and we can see these bursts of action potentials and the underlying synaptic potentials. And then how does this transform into a percept? So if one sees this stimulus that you can see here, probably you can see this black and white, or these black spots over a white background. And if I tell you that this is the color of a Dalmatian dog, then all of a sudden, probably this Dalmatian dog is going to pop out, and you are going to be aware of this. So your primary visual cortex is processing the same inputs, but then there is this interaction, in this case, with this prior, because you've seen a Dalmatian dog before, and there is a perception which changes. There is another example where one can see this stimulus, and if you see now a photograph with a banana, then the way of seeing these black spots over a white background changes. This is normally understood as an interaction, as an active process. It's an interaction between the information that comes from the outside world, and what is already, what you know, what's in your brain. So there has been a proposal of a theory of how this conscious perception occurs. So there are sometimes, for example, if you have had an audiometry done, they can give you these sounds, and then some of them are going to be below threshold. You may not hear them. Some of them are going to be above threshold, but still some of them, they may be making it through your auditory cortex. So the proposal made by Dehaene and Changeux in 2005 was the following. If you can see in this slide, there are some stimulus that arrive to primary cortices, and that eventually they propagate towards higher association areas, and some of them are going to reach frontal areas, and this is going to induce an ignition that is going to cause a reverberation in these recurrent circuits that we have mentioned before, and that this is going to be the base for the conscious perception. So the stimulus that would not make it through these frontal areas would not become conscious perception, but they would be subliminal stimulus. There is also an interaction with the fact that when someone perceives, normally we ask in order for them to report in some form. But this is the basis also for the global neuronal workspace theory by Stanislas Dehaene. And here you can see this paper in Science by Van Wacht et al., where, and I selected the model because it's more clear, but the data is very similar, how there is what in V1, which is the primary visual cortex, appears with a very similar response, but this ignition and this amplification is going to cause this nonlinear response, a much larger response that is going to be associated to the conscious perception of the stimulus. This has been reported with other methods like fMRI, like imaging of the brain. An invisible world gives this small local response, but a visible world gives this much larger activation. Or the same as you can see here, a non-detected sound remains locally in the auditory cortex, but a detected sound, as you can see, it activates many more areas in the cortex. So, I was working, as I said, I work in sensory perception, I work in sensory processing, in early sensory areas. areas. And some 15 years ago, I came across a technology which is virtual reality. And this suggested me that this was an amazing tool in order to study and to understand perception. So why is that? Well, let me tell you what is virtual reality, although now is quite well known, but just to be on the same track. So virtual reality displays in all sensory systems. Eventually, it provides multisensory information that instead of coming from the natural, from the outside world, is going to be generated by a computer, but it displays eventually in all sensory systems, enclosing the person in 360 degrees completely. It tracks the heads, the limbs, the body, and very importantly, is going to determine what we see by our position, such that it is very similar to what happens in the real world. And of course, there is stereoscopic vision and so on. And here you have a scene of the movie The Matrix, where people live in a completely virtual world. So therefore, one can have a multisensory environment where the afferences, the input, the information, the sensory information is now digital, is generated by a computer. And therefore, we have now an environment that is completely controlled by the experimenter. And this is why it's a very good tool to understand and to study perception. So we can think how much, it opens up many, many questions. So how much of what we perceive is actually out there, or how much is generated by our own brain, for example. And for example, this is an old environment of a virtual library where the people or the subjects of experiments had to walk around and do some tasks. And actually, this was a silent environment, but many people reported sounds. And this is why it's interesting to have a controlled environment, because then you can see how their brains are probably filling in this auditory information that would have been logical to have in a situation like this. So it's just an example to see how much of it is actually generated by our own brain. So we have this situation where we have a perceptual or multisensory consciousness of the external, in this case, virtual world, and also of our own body. So we have this consciousness of the world and of our own body, which consists of different senses: visual consciousness, auditory consciousness, tactile, olfactory, proprioception, if we are moving, temperature, and so on. Some of them, or ideally all of them, would be controlled by a computer. And this, of course, opens up many questions. So what is real, what is virtual, is actually what the world that we perceive is actually virtual. So our brain is the mediator, is reconstructing, and we know that there are many forms of energy out there that we are not aware of, and therefore we are reconstructing a part of it in our own brain. And then we have another aspect that you can see here, which is the representation of our own body, which is a very important part of the self. So how about self-perception? The perception of our body is actually a fundamental aspect of how we experience ourselves. And this is something that we started studying some years ago. We wanted to study to what extent we could feel a virtual body as our own body. So here we started exploring how visotactile correlations could induce ownership of a virtual arm. And how do we know if there is ownership, if this arm is felt as own? Well, we have first of all to ask the subject, but also we can measure it in different ways. We can ask the person where they feel their arm to be, or we can see how they respond to a threat, or we can measure with electromyogram how is their activity in their muscle depending of the movement of the arms and so on. This is another study. Here we found that not only visotactile but visomotor and proprioceptive correlations were also adequate in order to study ownership, to induce ownership of the arm. And here we can see actually that we can induce ownership of a full body. What you can see here is, first of all, first person perspective, visuomotor correlations. Someone sees themselves reflected in a mirror, and they have correlations also with the body reflected in the mirror. So all this together induces a strong sense that this body is your own body. And this allows to us neuroscientists to study the activity in the brain while this happens, to try to identify which are the critical areas and so on that are involved into this multisensory integration. So I have to say I don't have any feedback about how I'm doing in time. Well, so I'm near the end. I would like to just give 2 or 3 more ideas. One is that in studies that we have carried out over several years, we have seen that the brain has very high, is very plastic in the sense that can accept many different bodies as far as there is appropriate inputs, appropriate multisensory correlations. So I'll show you a few examples. For example, we can have one long arm much longer than the other, and still the ownership persists with some limits that are described in this study. One can also feel a smaller body, a younger body as own, like the body of a child. Or, for example, one can assume as own a body of a different race, a different color. And as far as I say that all these correlations are appropriate. So this is the important thing. The aspect, the visual aspect can be different and the illusion is not lost. So a very interesting aspect of this is that the shape of the body, the aspect of the body is going to have an impact on different aspects of how we process information. So for example, it's going to have cognitive impact, for example, on how we perceive the outside world. So there is a rescaling when the body is of a different size, or there is a decreased racial bias if we identify with a different race, or something that we have been working on quite a bit is how we can modulate pain by changing the aspect of our body. This is something that can be used in chronic pain patients. So I think okay, so I think I have time to finish what I wanted to say. I just got a message about this. So, so let me just finish going to 2 more aspects. I will just touch them very briefly. Maybe we can discuss later. So I have talked mostly about the importance on the sensory aspects of how we feel our body as our own, how are we conscious of our body, but what are the motor implications? Do we feel that we own, we control that virtual body? what is the effect of agency. So we know that if we feel a body as our own, one can have illusory agency. What this means is that if we feel a body as our own, like a virtual body, and this body moves, one sometimes can have the illusory agency over that body, like the feeling that one has generated that movement. And well, one can also control a virtual body by brain-computer interfaces. And we studied recently to what extent we feel ownership of the body. Once we feel ownership of the body, to what extent we can feel responsible by actions carried out by this body. And the motivation is in this slide here. Imagine that basically everything I have explained for virtual bodies, one can apply to robot bodies. We can feel also ownership over a robot body with some constraints. And what happens if by means of thinking, by brain-computer interface, we control a robot, as this is as the case is here, someone is sitting in Rome and controls a robot really far away. So we wonder to what extent we feel agent and responsible by the movements of external exogenous body. So we ran an experiment where we explored how people control a virtual body by thinking, by a brain-computer interface, and whether they felt responsible by actions, which was not seen well, but eventually that container over the table falls on the floor. And we saw that there is actually a relation between the activity of sensory motor areas and their responsibility. So I know it's been very fast, but just I wanted to give the idea that there are also motor implications. And finally, I can say that I think virtual reality is a useful tool to study perception, conscious perception of the outside world, of the own body, and it can be also used in order to do things that are incredible in the real world, like it is to be in the perspective of someone else, for example, or to do, and this is my last slide, something unthinkable, like it is, for example, to talk to oneself. So this is Dennett in our laboratory where he was scanned, he got his avatar done, and he could sit here and have a conversation with himself. So this opens up the door to do this kind of explorations of ourselves and to our consciousness. So with this, I finish. Thank you very much. And thanks to all the collaborators. I haven't listed them here, and also thanks to the funders.
Thank you so much, Mavi. Please stay with us because there will be, I think, a quite rich discussion at the end. And I now want to introduce the second speaker of this evening. And it's really my great pleasure to introduce Jean-Pierre Changeux, Honorary Professor of the Collège de France and at the Pasteur Institute Department of Neuroscience in Paris. At the same time also active in Kavli Institute for Brain and Mind, University of California, San Diego. I think that really Professor Changeux, with the discovery of the conformational modifications of the acetylcholine receptor, open a very avenue to understand the mechanism of brain plasticity and also many other possibilities. He, in his life, has used many different approaches, trying to utilize at the best different disciplines, from genetic to biology, to the imaging, to the pharmacology, in order to approach the complexity of the problem of consciousness. As you have heard from Mavi Sanchez, he with the Hene was the one who has produced this model of the global workspace neuronal workspace, which is one of the most important tentatives to try to explain the complexity of consciousness. His talk this evening will deal with the connectomics of conscious processing from the molecular to the cultural level. Jean-Pierre, it's up to you now.
Thank you very much for this introduction, and thank you to the Prada Foundation for giving me the opportunity to talk at this set of meetings on consciousness, and I am delighted to contribute to this topic by speaking more particularly to one aspect of conscious processing, which is the relationship between conscious processing and the connectomic organization of the brain, as you can see it in the right part of the slide. So this is a topic which is still ongoing, and I will present a few ideas and also some experimental evidence for these ideas throughout this presentation. And before going on this particular aspect, I would like to emphasize an important aspect of studying the brain is that it is, as you can see, it organized into several nested levels of complexity, from the molecular to the cellular, from the cellular to the circuits, from the circuits to more global processes in the brain. And that, after all, we are dealing here with some kind of system, which is, if I may say, a physico-chemical system, which is grounded on the molecular level up to the higher level. And the outcome of this complex organization is, of course, conscious processing, but also social life, cultural environments, and in particular, language, and of course, rational thinking. So it is important to have in mind this kind of organization with, again, hierarchical and parallel structures, and realize that all this complexity came out from evolution, and even there might be viewed through development, I will discuss later on, that all these levels derive from the other through some kind of Darwinian evolution, which are nested in a whole system, which conscious processing is the outcome. And therefore, one cannot relate conscious processing to only one top level, but remember that it is grounded into the molecular level, up and down. So this is, of course, an interesting feature, and I may mention the work of Tony and Edelman on some kind of measurement of this complexity, which is taking place through the phi parameter. And this idea was actually developed for the whole organism, and in particular for consciousness, but it's not, of course, unique to consciousness. Now, I will point three issues on the relationship of the conscious processing with the organization of connectomic structure. First, the paradox of brain humanization, then the architectures of conscious processing. Last, a very important issue, the postnatal epigenesis and cultural evolution. And I will close by selecting one disease and show how one could try to relate the molecular to the conscious processing level and its pathology with the case of schizophrenia. Now, what is the paradox of brain humanization? Now, one can simply follow the size of the brain, but it's also organization as a function of time in the ancestor of Homo sapiens, and see that from Australopithecus to Homo habilis, Homo erectus, and then Homo sapiens, there is a fast rise through only a few million years of the volume and complexity of the whole brain. So the dramatic increase of brain size and connectivity. And if you look, for instance, even at the level of Homo sapiens, you see that there is a relationship between erectus and sapiens, which is to some extent somewhat discontinuous. We have two species, and we have to understand how one may move from these ancestor of man into Homo sapiens. And I would like to say that from our present point of view, which is to try to understand conscious processing, of course, this evolution from, let's say, Australopithecus or some ancestor still living or branch of ancestors, like the chimp, there is an extraordinary development of brain cognitive abilities, and in particular language, self-awareness. We heard about it in Mavi theory of mind, and even so creativity, cultural evolution, art, science, and so on and so forth. So how can this change in brain organization lead to the development of such remarkable features and cognitive abilities? Now, the first thing that they believe in is that there are important genetic differences between the monkey and humans, or even between some relative ancestor of man, like Neanderthalensis to Homo sapiens. And in fact, this is completely wrong. There are the same small number of genes in mice, in chimps, and in humans, between 20 and 25,000 genes. So forget about the number of genes. Now, if you look closely to the sequence of brain proteins, and this was done recently by Guillaume Dumas in the Pasteur Institute. If you look at the sequence, then and their concernation, you can see that contrary to the testes, surprisingly, or even the lung, the brain proteins have a highly conserved sequence. There is not big change in the genetic organization of the protein determinants in the brain compared to many of the other organs. So, the sequence of the proteins which contribute to brain organization, brain structure, and brain function are highly conserved. So even there is a problem. And how to solve this paradox that there are indeed some change, they have to exist, few genetic differences having some important aspect on the connectivity and in the cognitive function, of course. So with Alexander Goulas and Klaus Ilg Tag recently, this is a paper which is in press in Cerebral Cortex, we propose what may be called a connectomic hypothesis, that how to try to explain that few differences, not in the structure of gene, but in the regulation of gene, which is of course under some genetic determinants, which are regulatory events in the genome, in the past few million years, lead to this important change in connectomic architecture. And the idea is that, in fact, there are change which we call connectomic fundamentals, which would be under the control of few gene regulatory events, and which would be especially under the control of these few genetic regulatory events, and have an important role in the development of brain complexity. So one simple thing to thought about is the size of the brain and the number of neurons, which indeed increase from chimps to humans. And there is certainly one connectomic fundamental, which is an extended period of brain ontogenetic development. Now, another interesting aspect concerns this multi-level organization I showed in my first slide. And there is clearly from the mouse to the monkey to the chimp to human an increase, scaling up of the multi-level organization of the connectome, which ultimately underlie enhanced conscious processing, which is engaged in the difference of conscious content, as Mavi mentioned, that you find in the brain of monkey and the human brain. Now, there is also another interesting feature which has been observed and demonstrated by Alexander Goulas, which is a shift or cortical layer reafference from lower to upper layer. You heard about the layer 3C, and which is going to introduce some long axon reafference. I will come back on that, compared to the layer 5, which is giving axon going down to essentially motor control and so on. So there is some kind of shift which increase the contribution of the higher cerebral cortex layers, which themselves, as you shall see, are assumed to contribute to conscious processing. Last, there is a very important consideration to take into consideration if we want to study adult consciousness, is that there is a considerable extension of the period of postnatal development which is taking place, and where the interaction with the outside world is contributing to the remodeling of the synaptic organization of the brain. And this lasts, as you know, about 15 years. And you have to remember that when, let's say, Homo sapiens appeared in Africa, the lifespan was about 30 years. So it means that when Homo sapiens was selected, it was selected as some kind of species which spent half of its life to build its body and mostly to build its brain. So this is an aspect which is extremely important to consider if we want to study consciousness, and in particular culture and the consciousness of the cultured brain. And I will make a special point about another fifth connection is connectomic fundamental, which is the interesting aspect of the development of the dendritic organization of the pyramidal cells of layer 3C that you see here, and especially in the prefrontal cortex, that there is a special dendritic expansion which is taking place beyond H2, and you shall see the importance I will develop about this. So it is clear that there are few events in gene regulation which are taking place, which for some of them are on the process of being identified. But in addition to that, there is this imprinting of the environment on the development of the cerebral cortex and of the brain in general, which I think is very characteristic of the human species. So what are the architectures of conscious processing? And before I briefly mention some definition, and which from the beginning I will stress that, as I said already, the human brain is conscious, rational, and social. And the social aspect of the brain, to my opinion, is under-evaluated in most of the studies done by the neuroscientists, who in fact study the brain of a single individual and forget a little bit about the fact that in fact human beings are at least as social as they are conscious. Perhaps they are even more social than they are conscious. So I will distinguish a few hierarchical levels of conscious processing, which was introduced by Mavi before: the access to multiple states of vigilance, wakefulness, sleep, dreams; the capacity for global integration, which I said at the very beginning of my talk is very important to globally integrate all the different components of the brain as a physico-chemical system, and the subjective experience will go with it. The capacity of social interaction with other agents, with self and other relationship, and the development of a culture. And perhaps another point which, if I may say, Damasio also mentioned, and which is essential for me, which is from Spinoza, the fact that not only in humans, but also in other species where consciousness is perhaps less developed, there is a spontaneous drive to survive, which was called conatus by Spinoza, the minimal form of intentionality. I will not discuss this aspect, but perhaps on the fifth day you have, will be further discussed. So there is a set of hierarchical level of conscious processing. And as I said before, we have to take into consideration seriously, if we want to understand them, that they set up progressively in biological evolution from simpler mammals and in humans through postnatal development. And these architectures are at this stage quite simple to classify. The states of wakefulness and sleep, which were nicely illustrated by Mavi in the previous talk. And there is a pontine ascending reticular activating formation here. This is from Damasio, actually, where there are cholinergic, noradrenergic, and glutamatergic circuits being involved. And this is a vertical aspect, I would say, the control of the cortex from the brainstem. Then there is the Thalamocortical Relationship, which was studied by Linas, and Paré and colleague. And this is, again, among the vertical organization of the brain and cerebral cortex. And now I wish to insist on the fact that there is another aspect which is essential for us to reach this globalization or this global control, which are the horizontal connection, which take place between cells in the cerebral cortex, between, I will say, such kind of columns or areas, whatever. And the issue was expressed by Stanislas and myself under the name of Global Neuronal Workspace. And we were, in fact, inspired from the work of Bars, which is a work strictly psychological, where he introduced precisely a global workspace where there are processors which inform and are informed by the global workspace. So there is a relationship between distributed processors and a global physical space for data sharing and broadcasting elements which then would become conscious. Well, if they are not broadcast, they will be non-conscious. So this global neuronal workspace, use the word global workspace from Bars, but in addition, of course, the most important aspect, which was not in Bars' presentation, the neuronal aspect of, and there we propose that, in fact, long-range axons, I already mentioned them, from mostly layer 3 of the cerebral cortex, would be involved in this horizontal relationship, which would combine with the vertical one, as it was actually proposed by Larcum recently, following these reasonings, as I said. So, what is this long-range connectivity? It came up from Ramon y Caral, who discovered the existence of these long-range axons, again from layer 2-3, and also from Desjardins, who was a French anatomist who discovered all these long-range fascicles of white matter, studied by Goldman Rakish, who was able to establish that, in fact, one may in fact, distinguish some kind of network between areas where these long-range axons are playing a critical role. And in fact, they are mostly abundant in the prefrontal cortex, but also in other association area, parieto-temporal and cingulate cortex, which would make some kind of workspace in neuronal terms, a global neuronal workspace in the brain. And you can see here, interestingly, that the recent work of Kennedy and Van Essen and others, where they try to look at the special network of the pyramidal cells in the cerebral cortex, they found that 24% of the strongest links are precisely coincidence with what we call the global neuronal workspace. So there is a, if I may say, strong anatomical support, and the main idea that you may see it illustrated here from a theoretical and now experimental point of view is that when sensory, for instance, the visual input enter into the cerebral cortex through the primary and secondary visual cortex if it does not reach the prefrontal cortex, which is itself connected into this global neuronal workspace with a reverberant aspect that you can see here on the right in this diagram, then we may have a way to view access to conscious processing from a non-conscious entry into the cerebral cortex at the level of the sensory area. So I wish to say that, of course, the idea of free entrant connection was mentioned by Jerry Edelman a long time ago, but not for consciousness. It was subsequently adapted by himself and by ourselves to conscious processing within the framework of the global neuronal workspace. But again, I mentioned Suzuki and Narkum, and clearly he here illustrates a scheme which is exactly what we proposed initially 20 years ago, that there are vertical and horizontal kind of connection involved in conscious processing. Now, the hypothesis we propose together with Stanislas is that, in fact, there is some kind of particular state of activity which is involved when, for instance, a sensory stimulus becomes conscious, and we call it ignition, which would be the physiological activity which would correlate with the subjective experience of conscious access. So this is still, of course, slightly speculative, because we see the ignition. We have some criteria for subjective experience of conscious access, but of course the relationship is going to be established. And in this respect, I wish to mention the work of Rolf Sema, which was also mentioned by Mavi, which illustrate that here you have a response, electrophysiological response at the level of V1, then V4, and when you reach the prefrontal cortex, then you have this ignition that you can also, of course, this was done early on with humans, and here have a distinction between this response which we relate to conscious processing and the non-conscious one in blue. And you may realize that the time scale here is interestingly slow, 100 to 200 milliseconds. And then there is this lasting activity, which is essentially present in the conscious thing and to a smaller extent in the non-conscious one. So this is not only true for simple visual stimuli, but also aesthetic one. And this is another talk about art. And here, the work of Kringelbach, who very interestingly was able to show that in the brain of the infant, through recordings that you can see here, you can find an ignition which is different when the child looks at this face and compared to this more agreeable to see one. So this is something which is interesting, and of course something to further investigate in the next few years. So there is some activity in the cerebral cortex, which Stanislas and myself consider as a signature of conscious processing of visual perception from simple stimuli to even, if I may say, cultural objects. So I would like to continue after this presentation of the horizontal connectivity which we consider as essential for the evolution of the content of conscious processing, to go to some of the other connectomic fundamentals I mentioned at the beginning of my presentation. First, the expansion of the multi-level structure, and which is interesting because this expansion precisely can be viewed as leading to the expansion of the global neuronal workspace that you can see here, which was just in lower animal existing, but much less developed. There is a dramatic increase of this connectivity, which from a theoretical point of view, give a more refined and abstract representation of the sensory stimuli, and which can be demonstrated actually clearly to have his number of errors in the reconstruction of a spatial location. And you can see here at the low level of the hierarchy is very high, and at the highest level, which is possible only in this highly expanded structure, is becoming higher. And you may contrast this difference between lower and higher level of organization through the, for instance, the working memory, which is for 1 or 2 items in the monkey, and in human of 7 plus or minus 2, as everybody knows. So there is a enhanced cognitive performance of the global neuronal workspace due to this connectomic fundamental, which is this expanded multi-level structure of the brain. Now, I mentioned already the shift of the size of the soma of the neurons from the inferior granular layer to the upper granular layer. And it's from the lower level to the superior granular one, which is indeed taking place a little bit in the monkey, but as you see here, become much more pronounced in the case of human compared to monkey. This is in pink, as you can see. And there is a very dramatic shift of the connection, which corresponds to, according to our view, to the increase of the contribution of long-range axons in the whole cerebral cortex and the whole brain, and in particular to the reentrant connection of the global neuronal workspace. Now, these are a few reflections I introduced with this early presentation about the connectomic fundamentals, which, as I said, may correspond to a few genetic regulatory events which do not change the structure of proteins so much, but for instance, the number of genes or the expression of genes through cis-regulatory mechanism, whatever, and said that one part of the paradox of the relationship between the, if I may say, the simplicity of the genome to complexity organization is the postnatal epigenesis and cultural evolution. Now, the new born brain is 5 times smaller in weight and in volume than the adult brain. So if you calculate how many synapses have to be built during this period from birth to, let's say, adolescence, you subdivide this 10 to the 14 to 15 number of synapses by the number of seconds. And amazingly, you find that per second, there are about 1 million synapses being formed in the brain of a newborn. So if you keep your baby in your hand for 5 seconds, 5 million neurons have been made up. So this means that there is a dramatic increase of connection which is taking place after birth, and that, of course, the long-range connectivity, which give access, as I said, to consciousness, is also increasing, as you can see here. So there is, even though the overall pattern of the long-range connectivity doesn't change too much, quantitatively, it's a dramatic increase in number of connection which is taking place after birth. And as I already emphasized, this is very important for letting the environment leave its imprint in the brain. And this is just to illustrate the relationship between the increase of long-range connectivity with evolution, and of course with the acquisition of language, as you see it from the work of Friderici. So the issue is how the environment is able to change the organization of this post-natal network. How is environment able to leave its printing into the developing network? And here is a simple scheme I proposed with Courage and Danchat quite a while ago, as you can see here, which I think is still true and very true, perhaps more true now than it was then. And the idea being that we have units within for instance, the cerebral cortex, where you have afferent axon and target neurons, and that there are stages which may be viewed as the critical stage or sensitive stage, where you need the neural activity to selectively stabilize the adult network. The experimental work of Hubel and Wiesel set the stage, but at that time, he thought that everything was predetermined. In fact, what you can see here, the idea is that there is a synapse selection which is taking place during this sensitive period, and that there is some elimination or pruning, using the American word, which is taking place during this stage where neural activity becomes necessary for the stabilization of a more older network. And we develop a model where we propose synaptic states, and the fact that the state of activity of the network, so this is for those interested in modeling, and that there is, in fact, a retrograde stabilization of the presynaptic terminals, and there are possibility retrograde factors which control this stabilization or elimination under the state of activity of a presynaptic body. And indeed, there are quite a number of cellular and molecular mechanisms which have been suggested for these kind of processes, which is a GABAergic inhibition for the opening of the critical period by Hensch. There are evidence with Rossi and the group of Pisa that the nicotinic receptor may even play a critical role in the development, and of course the neurotropic factor that Maffei, who is the head of this Pisa group, together with Tony Kawa, so beautifully illustrated. So there are indeed cellular and molecular mechanisms which are candidates to contribute to this selective stabilization of synapse, and which, of course, is taking place at many different stages of development and are nested one with the other during the postnatal development, including, of course, the long-range connectivity of the global neuronal workspace. Just one evidence I wish to show, which is rather recent, which is from the work of Rutazer, who simply shows here in case of the retinal ganglion cells in the tadpole, that you have a stimulation which is synchronous or not. And when you have asynchrony, there is no selection. When you have synchrony, you have selection and elimination of synapses. So there are extremely good proofs for simple system where this selective stabilization of synapse can be demonstrated. Now, of course, it's more difficult to carry this kind of experiment with humans, but I just want to show that if you look, and this is from the work of Bourgeois and Rakish, that synaptic density during postnatal development in the macaque, there is indeed after puberty and before there is some significant elimination, which is taking place. This is only the global envelope of all the synapses. And here you can see that in the case of human, there is a dramatic change, which is taking place. And in particular, the stage before puberty, between 2 and 5, for instance, years, and I will come back soon on that. So there is indeed both an extraordinary proliferation of synapses taking place postnatally, but also this elimination, which become dominant in the late phase of this postnatal development. Now, I mentioned this connectomic fundamentals 5, and just to illustrate, and this is from the work of Petanjek, who worked with Rakish, and it's extremely interesting, who look at what's happening in the dendrites very carefully in the cerebral cortex following the postnatal development of humans. And you can see that there is a very additional step, which is not seen in the monkey, for instance, at the age of 2 years and later. And the possibility that we like to emphasize that this is in fact one important connectomic fundamental where the language areas are developing, and through 3 and 5 years, and where, of course, the workspace capacity is sufficient for at 2 years and later to organize sentences in the language and reach higher levels of language development, which of course need an important contribution of conscious processing. So this effect of the environment on the organization of the brain, of course, is highly variable with the environment, and therefore we may consider that this epigenetic variability of the brain is at the origin of cultural diversity in the spoken language, in written language, in beliefs, in even some ethical rules, not all of them, but beliefs, within a genetic envelope which is common to the human species. So this is an interesting balance between this genetic envelope that I try to describe with the connectomic fundamentals and the gene which regulate this very few genetic events which regulate this evolution, and the development of this diversity, which take place not only for language, but for symbolic and philosophical system, and of course artistic styles. Now I'm going to close in a few minutes now on only one aspect of pathology. There was the issue of degenerative disease is absolutely essential problem for the future of humanity. We have to deal with it. Here I just want to select one case which is quite interesting for us. The relationship between schizophrenia and the global neuronal workspace. And this is from Martineau together with Dehaene, where they made some brain imaging between control and patients in the prefrontal cortex. And there is a very significant reduced activation of the anterior cingulate and frontal cortices in schizophrenics versus controls. Now, if you look at more precise cognitive tasks, which were done also by Stanislas in his laboratory, then you have conflict processing modeling task, conscious errors, modeling task difficult, and so on and so forth. So few complex perception tasks which are clearly altered, and there is a deficit in schizophrenics. So when you look now at the predisposition genes, you are faced with a problem because, again, there is no single gene proper to schizophrenia. There are hundreds of genes, and some of them are randomly hit by mutation and give a schizophrenic individual. But what I want to say is that among these genetic predisposition due essentially to simple genetic variation within the structural genes of some gene of the human genome. In particular, it was found by these groups that the nicotinic receptor genes, as you know, we have been working for now almost half a century on this nicotinic receptor. And the alpha-3, alpha-5, and beta-4 genes appear as some genetic predisposition to schizophrenia, and some of the SNPs in this gene, and in particular in the alpha-5 genes, give some increased risk of schizophrenia. I want to say that this is not the explanation of all schizophrenic patients and phenotypes, but at least a population of them. And and this was the work essentially of Uwe Mascos and Fanny Koukouli at the Pasteur Institute, created some modified mouse with a SNP of the alpha-5 subunit, which in humans cause the increased risk of schizophrenia. And very interestingly, they show that in the cortex and layer 2-3 again, there were some alteration of the nicotinic receptor, not on the pyramidal cell. Surprisingly, there are very few nicotinic receptors on this cell, but on the inhibitory cells. So it means that they contribute to what Mavi mentioned as the synchrony and relationship between the sets of cells and in particular the pyramidal cells. And that is the scheme that was published in this Nature paper 2 years ago. And here you can see that we feel that there is some kind of hierarchy of interneurons, which themselves regulate the state of activity of the pyramidal cell, which themselves give the long-range axon forming the global neuronal workspace, and as in agreement with what I just showed in the previous slide, create some hypofrontality, some decrease of the activity of the prefrontal cortex, as in schizophrenics. More interestingly, nicotine treatment have some effects on the mouse, and as you know, in the human patients, there is very often chain smoking in the schizophrenic patients, as if they were self-medicating themselves by smoking cigarettes. So this just to illustrate that we started with the top processing of alteration of conscious access, and finally we're able to reach the molecular level, even the molecular level at the level of a particular cellular distribution in the pyramidal cells of inhibitory neuron regulating pyramidal cell in the cerebral cortex. So we can conclude that there is here a bottom-up molecular modulation of conscious processing, which is taking place through the nicotinic receptor and its pathology. Now, I would like to close by one slide, which is inspired for the discussion I had with Paul Ricoeur, who was a moral philosopher who unfortunately left us a few years ago, and wrote a very interesting book, which is Oneself as Another, where he discussed something which I did not have the time to even mention, which is the social relationship between individuals, and in particular the way we can recognize in the other mind. I just mentioned the theory of mind, which developed like the sentence organization around 2 and 5 in the newborn, and this led us to discuss the variability between individuals in the human species, which is of course of great importance. There is of course variability at the gene level, and which still exists in human population. The work of Svante Pabo is extremely interesting in this respect, and in particular what the remnants of Neanderthal genome in Homo sapiens genome, and the distribution of these diverse genomes at the geographical level in the Earth. Now there is clearly individual variability of the connection in the case of what I refer to as the selective stabilization of synapse. Again, this is only a minor, mini scheme of what's happening at many places in the cerebral cortex during development. So there is, I would say, a variability which is essentially a socio-cultural variability associated with this selective stabilization, which is quite irreversible, not, I would say, not fully reversible, far from that. And if I have a bad French accent in English, it's simply because of that. And now there is also the variability of the conscious individual, taking care about, as Mavi mentioned at the end of her talk, the self of oneself, and of course all the societal and cultural memories of the cultural environment, which give the uniqueness of the conscious person which is exactly what we discussed with Paul Ricoeur a few years ago. So just to give the names of those who contributed to the work, Claude and Alexander to the connectomic hypothesis. Of course, Stanislas Anne, with whom we worked more than 20 years together in a very, very enthusiastic collaboration. Marina Picciotto, who discovered the first mutation, the consequence of mutation in the nicotinic receptor in the mouse. Sylvie Granon, Philippe Faure, and Uwe Mascos for these studies with the behavior of mice, which of course are very poor model for conscious processing, but at least give us some kind of information. So thank you very much. And my question is, do you like my brain? Maybe not. Thank you so much.
Thank you so much, Professor Changeux, for this very illuminating lecture. One point that has been touched by both Professor Changeux and Professor Sanchez-Vives was the importance of connections. Well, we like to be connected with all of you, so I think that you have the opportunity to be connected with us and send questions. At the right of your screen, you can find a text box where you can write your question or comments, and I will transfer your questions and comments to the two lecturers today. I may start because I have received already many questions, I have to say. So I will start with Mavi. Mavi, one question that has been arised is the following. Are the not detectable stimuli affecting any ways our consciousness, even if we are not aware about it, if they are subliminal? Do they influence in some way our behavior? And also, what about hallucinations?
Well, so there are stimuli that we have seen. I think we both have presented this schematic of the brain where there are some stimuli that stay in the primary areas without being able to activate the full recurrent circuits activating the whole brain. And as far, I mean, it's my point of view, but I would like Professor Changeux to I mean, he's an expert on this, and he generated the theory of the global neuronal workspace. But from my perspective, that I have worked and recorded from these primary cortices, as far as there is an activation in these primary sensory areas, and there is a response, and there is some response that it may have some propagation, even if it doesn't reach the frontal areas, is going to have some influence in the circuits. To what extent this is going to affect behavior, it depends. I mean, behavior implies activation of motor circuits and so on, but there is going to be, it's going to enter the activity in the cortex, and it's going to have some influence which can be local.
Jean-Pierre, you have been in some way asked to express also your view. What is your view about? So about the consciousness and consciousness.
Clearly, the preceding experience of the individual, for instance, during development, introduced traces into the brain, which in the adult are non-conscious, if I may say, in the sense that when you speak a given language, you don't even think that you are speaking French or English or whatever. So there is a deep influence of what is built in the connection of the of the brain during development, and also in a more transient way through a change of synaptic efficacies, which is such that what I may refer to as non-conscious stimuli may have strong influence on your conscious decisions, for instance. That's for me very clear.
Yeah, and Jean-Pierre, you mentioned how relevant, how fundamental is the process of ignition, is the process that in some way is able to activate in the workspace a specific approach and is determining a bit the selection of what is coming to our consciousness at each time point. So, and then the question is, which are the forces, the strength that move ignition, how ignition is activated in some way? Because, you know, ignition, as far as understood, is the one that activates the workspace in some way at a given time on a given topic. But what is determining the ignition process itself? What is determining this aspect?
Well, this is very simple and was mentioned by Mavi also. In our views, this is due to the recurrent processing. You know, you have a propagation which is not being modified too much from the, for instance, the visual area, primary, secondary. And then when you reach the entry in the prefrontal cortex and the stimulus become conscious, then there is an amplification of the signal which is taking place and which gives the ignition, as we say. So it's some kind of, not really all or none, but largely cooperative effect of the state of activity, which then is broadcasted into the workspace. And this is, if I may say, a definition of what a global workspace is. So the energy, there is nothing added to it. Is it intrinsic in the circuits? And when you reach this state of organization of the circuits, then if you go enough beyond the threshold, then you have this, I may say, explosive propagation, which is still under control and which is, as we believe, some kind of manifestation of an activity which is characteristic of the representations which are conscious versus those which are non-conscious.
Thanks. Mavi, this question is for you. Could the virtual reality also be used to increase empathy towards a different category of people, such as ethnic minorities, elders, disabled people?
Yes, so this is a very interesting area, and as I mentioned during the talk, something that we can do in virtual reality, which is very powerful, is to embody and to feel that our body is that of another person. So this allows us something that in the real world we can only do through our imagination. Some people are better than others at that, but the good thing in virtual reality is that without the imagination, you can just be in the shoes or in the perspective of another person, which is virtual, that you can feel embodied into that person, and then you can live through an experience. And so you can have a virtual experience, obviously, and experience it from the perspective of another person. And in our group, and also in other groups, there has been studies doing this, for example, by going into the body of someone of a different race, for example, and it has been demonstrated and measured in different ways that there is some identification, which is very automatic. It's not a rational, it's not something that one reaches by thinking about it, but it's like an automatic process, an implicit learning of how is the perspective of this other person, and there is an increase of empathy and so there is a change in attitude that can be measured, a change in behavior. And this can be done and is being explored by different people in order to be used to increase empathy and to understand the point of view on the experience of others, which can be, as it was mentioned in the question, people of a different age or people of a different group and so on.
Thanks. Another very important question to Professor Changeux. So you mentioned that the horizontal transmission, so what you call also data sharing in your global workspace hypothesis is a fundamental aspect. So if you make available to all the areas of the brain in some way and you share the information, you have really the possibility to even to produce the conscious state. So the question is this something that is telling us also that in the society we should use the same strategy, that is to share knowledge as it happens in our brain?
Of course we should. And if I may say, the past history of humanity have shown, in particular with science, that not only we can share information between individuals, between scientists themselves who make science, but also between the scientists and the more general population through technologies. And these populations benefit from the scientific knowledge. So there is not only this sharing which is taking place, but there is also an evolution of it, which you may discuss in the section about culture, because culture evolves. And in particular, science evolve and make constantly progress. Now the issue is, to some extent, now being reached that whether the technology that we are developing are going to contribute to the good of humanity or not. And this is a very, very important issue. And this reminds me of Pasteur, who asked us to do science for the benefit and for the good of humanity. It's sometimes difficult to know what is good, but certainly not to use this knowledge to destroy, to increase the poverty of people and so on and so forth. We have to be careful about what we do with this knowledge, which has developed creatively for hundreds of years. And in the next few hundred years, what are we going to do with it? I think it's a critical issue, in particular for environment, but also for the relationship between people in the world, and trying to have a more peaceful relationship between themselves.
Another very, I think, key question. The question is directed to both Jean-Pierre and Mavi. So, is brain plasticity a crucial element in favor of an anti-deterministic and anti-reductionist vision, or vice versa? Can the concept of brain plasticity and a deterministic conception of reality coexist?
I think it's an
quite challenging question.
Maybe you want to start and I will continue.
Well, let's start with Mavi and then you will to close.
Yes.
Well, I'm not sure I completely understand the question, but I mean, in brain plasticity, I don't see anything deterministic about it. So brain plasticity allows us to precisely to learn, therefore to change over time, to learn, to make changes, and therefore that plasticity allows to escape to some extent from what could be more deterministic, which is the genetics that one is born with. There is also the epigenetics. We have heard about this in the previous talk. And that's what I think, that precisely plasticity, which is triggered by external stimulation, by experiences, by our learning from the life experience, allows us to adapt and allows us to change.
Yes, this is, I agree with what Mavi said, because but this is based on the view which is current, that you have like Plato claimed that everything is determined by genes, if I may say, in the brain, and there is the empiricist view, which like Hume and Locke and Aristotle, that everything is learned in the organization of the brain, and the white sheet of paper on which experience write the organization of the brain. I think both of them are wrong. And we are actually in between. And as I said in my last slide, we have variability at the gene level, and we have also variability due to the plasticity level. And the question is the word deterministic. There is, to me, to my opinion, determinism in the relationship between the environment and the trace that the environment is living in the brain. So we may say, like Spinoza said, we feel free because we ignore the things which determine us. But what I would like to emphasize is that one of the main features of the human brain is precisely its variability between individuals with different cultures, but also within a given individual as a function of the states of the brain. And that's where reasoning and creativity is coming to precisely open the brain to variability, to innovation, and also to, as I said, the conatus, the ability to survive, which is characteristic of living organisms. So there might be some determinism for each of these individual aspects, but altogether, if you put them together, then it looks not theoretically, but practically very difficult to say this is what determines the character of this child and so on. Extremely difficult. And I would even say, even perhaps not feasible. And that's the reason why I make a strong difference between artificial intelligence and human robots, and robots, even human robots and robots in general, with human beings. Because the robots which have been built up to now have no culture. They have no experience that they did by themselves. compared to humans who, for 15 years, have a lot of experience with the society, with the culture, with the environment, and so on, which is such that under these circumstances, they build up their own personality. So whether a robot is or not is a person, I would say no, it's not a person. A robot is not a person for this reason, which I explain at the end of my talk.
So Jean-Pierre, I want to close this fantastic discussion with something a bit different. The last question is for you. You curated some major exhibition on art and science, and you are a member of the Scientific Council of the International Agency of Museum since 2007.
Yes.
Then, what is the role of a museum today from your point of view? What does it mean to be a cultural institution today? So we have with something that moves more in the direction of culture.
There is a statement, I don't know who said it, the people without memory are people without future. Les peuples sans mémoire sont des peuples sans avenir. And I think for me, the museum is stored culture and plays an absolutely critical role in the creativity and I would even say the survival of human beings. And this is something I may insist about because there is a tendency now to go to a cultural institution where you have only the last up-to-date views of singer, of art, and so on and so forth, and forget about Leonardo, Raphael, Michelangelo, or all Italian, actually, and Poussin or Cézanne. And I think this experience of the children with the art of the different historical periods of our society is, to me, necessary for the education and the harmonious organization of their brain. Now, I may close that there are cultural differences between civilization, as I said and mentioned. And I mentioned gave an illustration of the Occidental art compared to the Oriental art, just as an example, but I could take many other kind of examples. And which one is the best? None. All of them are important. And I think it would be extremely useful to have a deeper impregnation by this diversity of culture that we find, for instance, in our museum, or through education of people, to try to, instead of having clash between differences of culture, that to have a positive relationship, a constructive one for the future of humanity. And I would not dream too much, but I hope that this kind of relationship between the art of different culture should be a factor of reconciliation between human beings that we need a lot presently.
Thanks so much. Thanks so much. It's time to close. There was no better way to close than this recommendation. So Jean-Pierre, Mavi, thank you so much for having spent some time with us. And also thanks very much to all those of you who have attended until now. Please come back tomorrow. Tomorrow evening there will be, or tomorrow afternoon, or tomorrow morning, there will be a very interesting discussion about the imaging, structural and functional imaging, so relevant, as we've heard already today, to analyze consciousness. And we have two excellent, again, researchers, Professor Antonio Damasio and Yves Marden. So be back tomorrow, same time.
Thank you very
Good night.
much.
Goodbye.
Thank you very much.
Bye to everybody.
Goodbye.
Goodbye.
Thanks. Thanks.